Hey, welcome back. I'm Dr. Joel Duff, and today I want to talk about the mosquito that produced more than a little bite. It's a science communication story. I'm a scientist. I do biological research. I have a lab. I've had graduate students, master's students, and PhD students. And one of the things that I find is that students come in and they're all fired up about doing research, right? They want to discover new things in the world. And they're excited about that process. But the thing that I and my colleagues have to communicate to them is that scientific communication is very important. There's two aspects of scientific communication. There is communicating to your fellow scientists, all right, other experts in your field, and sharing your information, your data, the information you collect. You can't simply collect information for your sort of own curiosity and then sit on it your whole life. That's not really fair to the scientific community and the rest of the world and humanity, right? Things that you have found and that you have investigated deserve to be shared so others can learn from it. So one way to do that is to do that through peer-reviewed publications, through scientific communication at meetings and so forth. And that's a challenge for many students, right? They're interested in the science itself. They're interested in discovery. They find it difficult to communicate. And they're going to have to communicate because, I'm going to sit on their master's thesis committee or their PhD dissertation committee, and I'm going to ask them questions. And one of the important things they need to be able to do is communicate the knowledge they have to the rest of the community to convince us that it's worth giving you this degree, which is partially saying that you have not only the capacity to do science, but you should have the capacity to share your knowledge with others about what you have done so that they can either, do something with that data, trust that data, repeat the types of things that you have done. And to do that, you have to communicate faithfully, right, and accurately. The other form of scientific communication is communicating the importance of what one has done to the general public who may not have not to be as intimately familiar with your particular research area and your knowledge. That is often the much more challenging aspect of doing scientific research. And not every scientist has to be able to do that. I think it's important that they all have some skills in that area, but you can hide away in a hole, do your research, write it up in scientific publications, allow others to communicate your information. And there are scientific writers, right, that take complicated concepts and try to communicate them to the general public. All right, all that's a preamble to say that I just want to tell a little scientific story, a story of scientific writing and communication. And I'm going to tell that through I believe this is a 2013 paper that came out in PNAS, and it involves the picture you see in front of you, which is a fossilized mosquito from the Montana area I believe this is around like 30 million year old rock. In fact, this paper discusses the results from 36 different fossil mosquitoes that were found. But this particular one is of fairly, let's say it made a splash, or as my title says, it created more than a little buzz in the scientific communication world at the time, and it was picked up by a number of different popular news outlets, and so some people may have heard of this particular mosquito. So what I want to do is I want to show you the headlines. All right, I want to show you the headlines from popular press. I'm going to show you what did the headlines say, and then from that I want you to get an idea of like what do you think that they found from reading those headlines. Because that's what most people see today on social media, right? On Twitter or Facebook and so forth, they might see the headline and they walk away from that. We know that the click-through rate on these types of articles is very low. And the headline essentially is what the takeaway message. And so what is the takeaway message from the popular press articles in 2013 about this particular fossil? Let's take a look at those. So here's a couple. And when I say popular press, I'm just going to call these news outlet responses. They're not scientific papers or journals. They're reporting on this, and they're not necessarily scientists reporting on this. So how do people who are writing for these types of outlets just sort of communicate what they think was in this PNAS paper? So let's read a couple of these. Blood cells found in fossilized mosquito. This week, a droplet of blood from a 40 million years ago was discovered, frozen in time, inside a fossilized mosquito in a riverbed in Montana. Mosquito fossil with blood-filled belly discovered, the Japan Times. Unprecedented blood-filled mosquito fossil raises questions over evolutionary dating methods. Now, this comes from CNN, not that CNN. This is the Christian News Network, which is very sympathetic to what we'll call young Earth creationism, a particular perspective on how to understand the chronology of the world and a young Earth. They're very skeptical of evolutionary biology and old age ideas, and so they're definitely putting a spin on the meaning of and the significance of this particular find by saying that this blood-filled mosquito questions evolutionary dating methods. And I mentioned in the preamble that this is like a 30 million year old mosquito, fossilized mosquito. Blood mosquito fossil supports recent creation. So they're from Institutes for Creation Research, again, a young Earth creationist organization that writes articles and has a big footprint in terms of people seeing their information on social media. Bloody mosquito supports recent creation. Fox News, rare blood-engorged mosquito fossil found. And first fossil mosquito found filled with blood from Answers in Genesis, another young Earth creationist organization, to which hundreds of thousands, if not millions of people would have seen that particular headline splashed across their webpage and various social media outlets. All right, so what's your takeaway from, if you don't know anything about this particular fossil, and these are the headlines you see, what do you think that they found in this fossil? Right? Blood engorged, blood found, right? They found blood inside of the abdomen of this particular mosquito. I don't know about you, but when I saw that, I'm skeptical of science reporting, and so I have a natural skepticism against all sort of popular lead titles, and because I'm constantly let down by them, right? It's like, oh, that sounds really exciting, and then you go read the article, and maybe the article is still kind of exciting, but you always find that what your first impression is not actually what was found. All right, and that's going to be the case here. They didn't find blood cells, just to give away the story. They found evidence that suggests blood was there inside of the fossil abdomen. Nonetheless, these particular titles certainly give you the impression that it's almost like, right, the Jurassic story type of thing. Hey, we found this fossil of a mosquito, but there was blood inside it, blood gorged, right? And so maybe we could actually extract this blood. Maybe there are biomolecules like DNA that could still be there, and we could like recreate a mosquito from 30 million years ago. Now, I don't know why anyone would want to do that, right? It's not like making a dinosaur, which is what everyone wants to do, is make a dinosaur. But nonetheless, the same idea that somehow there's like almost fresh material like oozing out of the abdomen of this mosquito in this fossil. So here's a couple, I'll say, scientific journals, popular journals. So not reporting the original research, but taking original research and then writing it in a way for the public, usually a curious a public that's curious about science, right? So Scientific American, right? I'm a subscriber to Scientific American. Their title is "Fossil Mosquito Found with Belly Full." Hey, belly full of what? I don't. So then it kind of. But when you read the first line in the article, they state that the fossil contains iron in the fossilized belly of a mosquito, indicative of its last likely meal of blood. Now, I'd say they found iron there, right? They measured the presence of iron in a specific location in this fossil, which is the abdomen. which then you can use to infer that there was blood inside this mosquito. You said, like, well, whoop-de-doo. I mean, wouldn't I expect that? Well, not necessarily. I mean, this is a fossil from an ancient mosquito. There are very few fossil mosquitoes. They're very difficult to fossilize and hard to find. And so we don't have any evidence from mosquitoes from long ago, whether they actually sucked blood from other organisms or whether they had different types of ways of getting energy or completing their life cycle. Because remember, it's only the females that suck blood, at least females today. And then you could ask the further question: is it females in the past, only females in the past, or maybe males and females both use blood in certain species of prior mosquitoes? And in fact, this paper actually addresses that because they looked at 36 different fossil mosquitoes, and some of them were male and some of them were female. Off the top of my head, I'm not exactly sure how they can tell which ones are male and female, but they determined whether they were male and female before doing their analyses. And what they discovered was the female ones that they identified as being female have this signal of having iron in the location of their abdomen, whereas the males didn't have that same signal. And so that suggests that the present is very similar to the past with respect to how mosquitoes operated. Also from The Scientist, not Science Magazine, but The Scientist, which is another journal just similar to the Scientific American. They say fossilized mosquito blood meal is their title. And they begin by saying a 46 million-year-old female mosquito. Oh, forgot this particular fossil. The one I showed you is 46 million years old. There's kind of a range of different ages. Contain the remnants of the insect's final blood meal. Right, this is more accurate reporting, saying more about what they're saying rather than giving the impression that they found fossilized blood. Which for most people, when they hear blood, they're thinking blood cells, red blood cells, as if red blood cells. Does this sound at all familiar? You've heard the stories about, Mary Schweitzer and her discovery of blood cells or things that are blood cell-like that are found in dinosaur bones, along with soft tissues. We use that kind of language, and even the scientists who discover these things use language like that, although they're actually qualify their language. If you read their articles, it's soft tissue-like material, not that that's the original material that's still present. It's still highly modified, as we're going to see in just a minute. I'm going to show you what modification means. They're not saying that, you cut it open and it's like, I just sliced open, a fresh bone, and the tissues that are there exactly like the tissues that are present in today's animals. They're extremely modified and altered, but they still have a similar, they can have a similar texture to them without being the same thing. In this case, we find that, hey, there's evidence that there was blood inside this belly, and that blood is, what is it? It's the remains of what was in blood. They give us enough evidence that we can say, yes, blood was there, but all the components of that blood aren't necessarily the same as that blood. I mean, they didn't crack open this rock and blood ooze out of this mosquito's belly. All right, let's go to the research paper itself. Right now, imagine that that scientific communicators who, or like a student, right? A student of mine writes a paper and they want to communicate this to the public, and they just use the title of their paper. Because remember, the title in a scientific paper is trying to communicate most accurately something important about what's in this paper to the scientific community, to others who probably have similar expertise. And you want other people to read it. And so in a way, there is some form of salesmanship to writing a title, but it should still be, there's an emphasis on it. It should still be fair. It should represent what's in it. You shouldn't be disappointed when you read the article. Like, oh, I was led to believe this, and now I find out, it's really not that. Right? It's not quite what you said it was in the title. So I think this is a decent title. But again, this is not what you would write in CNN or Fox News, right? Hemoglobin-derived porphyrins preserved in a Middle Eocene blood-engorged mosquito. Now, the blood engorged mosquito part is actually a little bit of that's an inference, right, from the evidence that they find, that they infer that this mosquito must have been engorged in blood because of the amount of material of iron, and we'll see porphyrin, what's called porphyrin, which is mentioned right here in the title. And so this is a fairly, I would say, we'll call this a descriptive title for this particular research. All right, I'm not even going to show you any more of the paper because it's profiles of molecules through mass spec. I think it's time of flight mass spec. So a very sophisticated set of chemical analyses. And the pictures aren't worth 1,000 words to anybody but somebody who knows exactly what that particular mechanism is. Nonetheless, it's compelling evidence that abdomen of that mosquito in the fossil record at one time had a lot of blood in it, right? Blood of some kind of vertebrate. Great. But now we have this issue but here's how it was presented to the lay audience, all right? In this almost Jurassic Park-like way. Like, this is really exciting. And it leads people to believe that, like, oh yeah, we're finding like this blood, we're finding like ancient biomolecules that potentially we could read or do something with from these ancient organisms. We're not really, it's nothing like that. Nothing really even close to that. The scientists were just interested in how do mosquitoes behave in the past versus how they behave in the present, looking at how they may have changed their habits. Now, so in PNAS, Proceedings of National Academy of Science, it's a very prestigious journal. And you don't have much space. And so it's just like you get right to the point. You better stay right to the facts. You have a short discussion section. There's no flowery stuff in there, at least not supposed to be. It's just communicating, here's what we found. Here's why it's important. And here is our materials and our methods and how we did it. For articles that PNAS believes will have some general public interest and they can see that, oh yeah, people are going to find this interesting or maybe even misread this, they often will they'll invite somebody to write a kind of a review or a perspective on the particular paper. Like, how does this paper fit into the bigger topic of something? And that's what they did. They had Derek Briggs, a well-known paleontologist, write this cover article basically at the front of the journal saying, a mosquito's last summer reminds us not to underestimate the fossil record. And his point here is that you can crack these thin layers of shale from the Green River Formation open, and under a microscope even you can find these very, very small fossils, which is like, okay, it kind of looks like a mosquito, but what else can we do with it? We're finding that we can discover a lot more than we ever imagined we could. Right, 20 years ago, we never imagined we could discover something by reading the biomolecules that are left behind in these fossils that have been preserved. So this really is a very interesting paper from the perspective of the technology has gotten to the point where we can identify specific molecules that remain in fossils. And from those molecules, we can learn something about the past. In this case, we can identify that blood was there because of the type of molecules that are left behind. That's not the most dramatic finding, and so this is a general summary. And what I'm showing is the main figure from this. And it's going to get my other main point across about the actual research here. And I want to apply this to how this particular research has been mishandled and misused and misunderstood by certain members of the general public. All right, so let's go to, hey, here's a modern mosquito. All right, female mosquito. Sucking up blood, and blood has all vertebrate blood, has a molecule called hemoglobin. It's a really complex molecule, protein. And proteins are very complex biomolecules. There is this, in hemoglobin, there's a portion of hemoglobin that is this called the heme molecule. And it is structured such a way that it holds on to or contains this iron element. And that iron then is attracted to oxygen, which then helps the oxygen bind to this particular spot, and you can carry oxygen around your blood. So that's a really critical molecule in your blood and in your blood cells, and bound to your blood cells. And you've got a lot of it, right? So if you have blood, you've got a whole lot of this heme molecule that's helping you carry your oxygen. Now, in this fossil here, what they're saying is, look, hemoglobin is this really complicated biomolecule of proteins or amino acids. Those amino acids are all decayed away, right? They've broken apart and they've been lost, to time. So over time, the protein breaks down, but these portions of the heme portion of the hemoglobin forms an extremely stable molecular bonds around this iron molecule. And that particular product is called a porphyrin, right? And so when it breaks down, you end up with what is called these porphyrins, and they're a breakdown product of hemoglobin. So it's sort of like it's unstable, and that instability causes the bonds to break, and eventually the individual elements are lost. All right, or kind of replaced and pushed out by the fossilization process. But the heme molecule or the porphyrin is so stable that it stops degrading at some point, right? That it reaches a point of incredible stability in which it can last for hundreds of thousands of years, if not millions of years, in exactly that state, and it's not going to break down any further. Maybe in a billion years, all right, hundreds of millions of years, it's going to maybe eventually break down under just the right conditions. So for example, in oil, right? Oil is a biomolecule. We acknowledge that biomolecules can last for long periods of time, despite what some people might kind of suggest to you is like, oh, all biomolecules, decay quickly over time. Well, yes, most do, but there are some states of biomolecules that are extremely stable, oil being one of them, right? So, and in oil, most forms of oil, crude oil, if you look in it, there are small amounts of this molecule called porphyrin. All right, so what was found in the fossils? Not blood cells, right? The blood cells are long gone in terms of the molecular structure of all the other components of the blood cells. But what's left is this breakdown product, which reached a point where it was very stable and it stayed localized in this particular area of the abdomen. So other portions of the abdomen are being compressed, converted into there's probably some type of biomolecule that's also slightly present there as well. But it's also being replaced by other atoms that are inorganic. But leaving the general shape so you can see, like, here's where the abdomen of that particular mosquito was. But in this region, there is this porphyrin, which remains stable in that particular localized region. And now we've gone in and we can actually pull that biomolecule out and say, look, there's still that biomolecule left. All right, so it's not nearly as sexy as the original headlines would suggest, right? There's not blood here. We're not sucking blood out of a fossil. We're certainly not finding DNA in this particular fossil. DNA is a lot less stable than hemoglobin. But you shouldn't get the impression from these reports of fresh tissues or cells that are found as if the cells were exactly as they were when the organism was alive. They're highly modified, as this particular figure shows. Let me transition to talking a little bit about how this relates to the young Earth creationism story. So as you saw on those, let's go back here real quick. As you see in these headlines, bloody mosquito fossil supports recent creation. First fossil mosquito found filled with blood. So, where's the fault in terms of the communication here? It's a two-way street. All right. Scientists, if they're not involved in their own science communication, and being very careful about how they, not just selling a story to the public, which is what happens a lot of times. They get caught up in selling the importance and making some kind of connection. And you see in my blog post, I have to write something at the beginning of a blog post that sort of like is a catch, right? Why is this interesting? And I'm always thinking to myself, like, I want to be honest about the catch. I don't want to be like, oversell the product such that you get 3/4 of the way down through the article and it was like, well, I mean, this is really not nearly as important as what you made it sound like. So it's hard to do. Effective science communication is really difficult to do. But it's really important, because if you don't effectively communicate, you create misconceptions. And those misconceptions grow over time. And unfortunately, misconceptions may not just be misconceptions. They may be intentionally misconstrued and used by, let's get straight to it. The Institute for Creation Research and Answers in Genesis, in a way, they are, as creationist organizations, they know that their audience, their knowledge of science, most of them, comes from reading this kind of material up here, right? These kinds of headlines, especially since they read a headline from Creation News Network, which says, unprecedented blood-filled mosquito fossil raises questions over evolutionary dating methods. There was nothing in that original article that suggested that they were doubting evolutionary dating methods, that their research was questioning that at all. That's their inference. That's inference. That's their taking that particular fossil and saying, oh, look, they found blood in there. Well, blood can't survive very long. Anybody knows that. If you take your blood out and put it on a shelf, right, put it on this table, it's going to decay over not a very long period of time. And so blood cells, they're really fragile. And so if they found blood in a fossil, then how could, this fossil be said to be 40 million years old? That's utterly ridiculous, right? And therefore, that whole evolution thing, which they equate with just as a catchword, as a trigger word for they believe in, don't believe in the Bible. They're using the audience's impression from these particular headlines that there was actual blood found in this particular fossil. Now you could ask the question, did the person who wrote this article from Christian News Network, did they read the original paper? Right? I'm gonna say they probably didn't read the original paper. What they've probably done is they've probably read, whoever wrote this paper probably read these papers, right? And those papers, if you read them correctly, I've read all of these. Some of them will actually, embedded somewhere in there, kind of talk about the evidence, the actual evidence that was found, which is the heme molecule, the porphyrins, and they also looked at the amount of total iron that was found there. They might not even understood that they're not talking about the actual cells, because the articles make a big deal about how this proves that there were blood cells there. Didn't say they've proved, if you read it super carefully, it doesn't mean they've proven that the actual blood cells are there, but they're saying that at one time blood cells were in this particular mosquito. But it's very easy to go from that to, the third person who writes an article based on someone else's review of an original research article, which might even be 2 steps removed from the original article, because there's a place called Science News who summarizes science news articles. And they're usually fairly accurate, but they're still kind of sensationalistic in the sense. And so if you don't go back to the original article, it's very easy to lose track of the real story. And so you end up with headlines like this, which may or may not be attempting to manipulate an audience, knowing that they're not really telling the truth. They may just not really understand the story themselves. And this is a natural inference for them. It's like blood. You can't have blood that's 40 million years old because it will decay. But as I've just shown you, if you go back to the original article, it doesn't say there is blood there, right? They're saying there was blood there a long time ago. And the process of decaying that protein, the hemoglobin protein, is not something that would happen just instantaneously. That in itself might have taken hundreds or thousands of years. And to get to porphyrins, to get to that very stable state of a hemoglobin molecule, of the hemoglobin turning into the porphyrin, might have taken a very, very long period of time itself. So it's not suggesting that the world is young. Like Institutes for Creation Research, fossil supports a recent creation, or first fossil mosquito found filled with blood. I mean, Answers in Genesis, it just says right out, found filled with blood. Completely untrue. But this fits right in with their perception of the world, right? If the world is only 6,000 years old and all these fossils, like this mosquito fossil, was deposited only 4,000 years ago, in their mind, just 4,000 years ago, and quickly preserved in a global chaotic flood, it could still have blood in it because that's not that long ago, and it's been just preserved for that period of time. And so I think they may read something like this without reading the original research, which they often don't do before they start talking about these things. It fits what they already believe about the world. Their misperception of the article fits their perception of the world, and so they don't feel the need to research it any further, right? It's like, well, confirmation. It's like blood. Ancient blood, and these crazy old earth, people think that this blood could last for 40 million years. That's just completely nuts. Yes, science communication is very important, and I don't have the answers as to how to do it, other than it takes work. It takes real work to consistently communicate difficult, complex stories and put them in simple terms. Converting something to a simple term without actually creating misconceptions like this is really hard. All right, I'm babbling on now, and so I think I better wrap this up. Really interesting fossil. I mentioned that they found males and females, and the males and the females were consistent with their prediction of what they would find inside the abdomen. These are ancient, decayed products of what was a larger biomolecule in a living organism at one time. And the beauty of these types of new techniques to look at these molecules is that we're going to be getting a lot more information out of what in the past somebody would have just looked at and kind of like tossed aside. It's like, we can't really learn anything from this. Oh, there was a mosquito and it was alive in this particular time period. That's all we could say before. We're going to be able to say so much more in the future. That's a story of science communication. I feel like I've not even done a very good job of communicating this particular story about science communication. Science communication is hard. It takes practice. It takes work. And it takes an audience willing to spend more than the time it takes to read a headline to understand a story. And so that's the difficult thing we're trying to, I think all of society is grappling with, is that we get our information in little bite-sized chunks. And science isn't a bite-sized thing that you can learn about. If we learn anything from this coronavirus stuff, it's that anyone out in front who only has a moment to make a point is going to find that it's hard not to leave misconceptions in people's minds, or it's hard not to allow them to take what you've said and read into it a whole bunch of different things, because they're unwilling to read the background information as to why different conclusions are reached. All right, that's it for me. Thanks for listening, and I'll be back sometime with more stories from science and otherwise. Thanks a lot. Bye-bye.